Ladies and gentlemen, dear colleagues, I'm delighted to be here today with you to open this special session dedicated to challenges and opportunities in HIV science. I've had the opportunity yesterday to express my personal commitment and that of the French government regarding HIV and AIDS control. To meet this collective challenge and end the epidemic in 2030, researchers strive continuously toward new paths. This is precisely the subject matter of this special session. I don't think I have to present in this audience, and generally worldwide, Dr. Anthony Fauci, who has kindly accepted to talk about sustained, art-free HIV remission, opportunities, and obstacles. I just want to emphasize that Dr. Fauci is since 1984 the director of the US National Institute of Allergy and Infectious Disease. In his capacity, Dr. Fauci oversees an extensive portfolio of basic and applied research to prevent, diagnose, and treat established infectious disease such as HIV/AIDS, tuberculosis, and malaria, as well as emerging diseases such as Ebola and Zika. Dr. Fauci, you have advised 5 presidents and the US Department of Health and Human Services on HIV/AIDS, and you have been one of the principal architects of the President's Emergency Plan for AIDS Relief, a program that had saved millions of lives throughout the developing world. You have also been the longtime chief of the Laboratory of Immunoregulation and made many contributions to basic and clinical research on the pathogenesis and treatment of immune-mediated and infectious diseases. It is my great pleasure to attend to your lecture. Please welcome with me Dr. Fauci.
Thank you very much, Madam Minister. It's really a great pleasure to be here with you this afternoon to talk about in this special lecture this extremely important subject. And that is one that has been greatly discussed at this meeting and before, and that is the possibility or not of sustained ART-free HIV remission, the opportunities and the obstacles. So let me just give a bit of a background for why we're even talking about this. First, I want to disclose that I have no relevant financial relationships. Now, we all know that there has been extraordinary success and efficacy in the use of antiretroviral drugs to suppress HIV viremia, as shown on the left-hand side of the slide, which, all other things being equal, is the usual scenario of an infected person being put on ART, achieving full suppression of their HIV viremia. This has led, over the last several years, since we've been broadly practicing this where it's been available, that the original life expectancy of an HIV-infected individual in the early 80s could be measured in a year to a year and a half. If you fast forward to where we are today, if a 20-year-old comes into the clinic newly infected and gets started on an antiretroviral regimen of 3 or more drugs, you can honestly project to that person that they would live an additional 50-plus years. And so this is truly a success story. Having said that, with drugs that work so well, why would anyone want to be off those drugs? And the answer is here, because there are a number of issues that are the rationale and motivation for pursuing an ART-free sustained remission. The already recognized ongoing drug toxicity, the uncertainty regarding the possibility of unrecognized long-term toxicity, the issue of pill fatigue or not wanting to take a pill every single day for the rest of your life, the stigma associated with taking ART, and of course, the cost of long-term ART. What is the stumbling block to an ART-free remission? As we all know now, it's the HIV reservoir, which was discovered in the late 90s by Bob Silicano and Taehyuk Chung, reported in Nature. We're all very familiar with that. When cells get infected, the overwhelming majority die, but a small subset then go to a latent resting state, which is the recalcitrant reservoir that defies eradication. We have tried, as many others have, and this is a paper from my lab in 1999, to try and empirically stop therapy in people who've been on therapy with complete viral suppression for 3 or more years. And as you can see, sadly, in this picture, that essentially every single one of them rebounded to their original viral set point. And so we came to the conclusion, as I articulated in this article many years ago, that the latent reservoirs are the true obstacles to the eradication of HIV. And so what I want to discuss with you over this period of time are the 2 pathways that have been pursued to attaining a sustained ART-free HIV remission. The first is an attempt to actually eradicate the replication-competent reservoir, what we refer to as a classic cure. There have been a number of strategies that have been pursued in this attempt to eradicate. The first is the one that has been most aggressively pursued, namely to activate the cells in the presence of ART and to so-called flush out or shock and kill the reservoir. As a matter of fact, we tried that many years ago in the late 1990s. And what we did is that we used in an individual, several, who were very nicely, durably suppressed with their viremia, we gave them multiple infusions of interleukin-2, which is as powerful as any T-cell activating agent that has been used. We thought, somewhat naively, that we may have eradicated the reservoir because we looked and we could not find replication-competent virus when we cultured more than 300 million peripheral cells and did multiple lymph node biopsies. Yet, to our disappointment, and a forerunner of what we would see later on, when we empirically discontinued therapy, the virus rebounded in everyone, including these 2 representative patients. The virus is in red. The size of the reservoir is in the blue bar. This was the beginning of an arduous journey, because over the last several years, as shown on this slide, which is not complete, but represents many of the attempts, all of these have failed to truly eradicate the reservoir. So I take away from this some lessons or caveats in my now many-year experience in trying to do that. Merely decreasing the size of the reservoir is insufficient. It must be eliminated. And we see many reports, including at this meeting, of someone or some group that decreased the size of the reservoir. Even if you cannot measure a replication-competent reservoir in the blood or lymphoid tissue, it does not mean that it is not there. I have sad and disappointing experience with that. The only way to establish that you've eradicated the replication-competent reservoir is by interrupting ART and waiting a considerable period of time. Other approaches have been immunotoxic therapy, various versions of that. I'll give you an example of one of those that is being considered. It's bispecific T-cell engagers or dual affinity targeting, where you link up a cytolytic T-cell to an HIV-infected CD4-positive T-cell, a variety of other approaches that are in vitro proof of concept, but have not yet been used, certainly without success, in the human model. And then we get to the issue of stem cell transplantation. We're all very familiar with the now iconic Berlin patient, Tim Brown, who received a stem cell transplant from a donor homozygous to CCR5 delta 32, he already had acute myeloid leukemia, so there was a reason for him to get a bone marrow transplant to begin with. In this New England Journal of Medicine paper from 2009, already it was 20 months and no rebound. But then, if you go many years forward, this very likely is a cure, as Tim, now years out, still has no indication that he has replication-competent virus. So if ever there was a cure, this would be one. But let's drop back a few steps and take a realistic appraisal of the role of stem cell transplantation in the cure of HIV. I believe it is a reasonable approach for HIV-infected individuals who require stem cell transplantation for the treatment of leukemia, lymphoma, and related diseases. However, the risk, morbidity, and expense for the average HIV-infected individual who's virally suppressed on ART outweighs the potential benefit by far. This is not scalable for tens of millions of infected people. But I have a thought when I was putting this together. If we could maintain an HIV uninfected person who happened to have leukemia in complete long-term remission without necessarily curing them, by just giving them a single pill every day, would we be considering stem cell transplantation in them? I think we should consider the story of Gleevec for chronic myeloid leukemia. We're not doing stem cell transplantations in those patients. And then there's gene editing. Very promising. In fact, this is something, and this is just one example from the New England Journal from Pablo Tebas and Carl June, talking about gene editing of CCR5 in autologous T cells. And now, with the advent of CRISPR-Cas9, this indeed has some promise. But if you look at the experience of trying to eradicate, I can come up with, I believe, for now, this statement, that complete eradication of HIV from an infected individual, for certain, will be extremely difficult. At best, at worst, it might be impossible. But I'm a cautious optimist, so I don't say that. But at best, it will be attainable in only a small portion of the infected population. So let's move on to the second, which I believe is more realistic and which has been the subject of a lot of discussion at this meeting and will be. And that is controlling viral rebound without necessarily eradicating HIV after you discontinue ART. I use the terminology now, sustained virological remission. So those of you who know me know that I have been staying away from using the word cure, and I prefer to use the word sustained virological remission. So here is the matrix that I build upon when I talk about this, and I'd like to go over and share it with you. The common denominator in the best possible world would be the early suppression of HIV viremia with ART. Why would that be important? Several reasons. One, we know from studies from a number of laboratories that that limits the size of the reservoir. Second, it may favor the establishment of an unstable HIV reservoir versus a stable, more recalcitrant reservoir, as Louis Picker has described in his CROI presentation last year. Next, it favors a global immune function, namely preservation of immune function. It also might favor the development of HIV-specific immunity, what I refer to as the live attenuated vaccine effect. What do I mean by that? In malaria vaccinology, a promising approach is to infect someone deliberately with live malaria parasites, followed immediately with chloroquine therapy before they become symptomatic. Now, obviously, we're not talking about vaccination now. We're talking about someone who's already infected. but therapy quickly could have this effect. And also, there are potent ART combinations today, in 2017, that can more rapidly and effectively suppress virus replication. So with that as an accepted best-case scenario background, let's quickly march through some of the options. The first is, what about natural HIV-specific immunity without any other interventions? I already showed you this slide that at least back in 1999, when we took people empirically off therapy, they all rebounded. If you look historically and go over the literature, you find that the time from stopping ART to rebound, and look at people who were started on therapy in the 1990s through 200, the median time to rebound was 7 to 14 days, a la the slide I just showed you. When you started therapy from the 2000s until most recently, the median time to rebound now is about 28 days. In fact, here is some real data from my own lab in which we looked at a group of people who were empirically stopped from ART who had long-term suppression of virus. And as you can see, it's variable, but the median time to rebound is about 28 days. Now, there is an extraordinary spectrum that we've been discussing at this meeting and will continue to discuss, namely major variations in differences in time to rebound among people, individuals treated early in the course of HIV. We have the Mississippi baby with 27 months. We have the French teenager with 11 years. We have a subgroup of the Visconti cohort that's anywhere from 4 to 7 years. We have the Spartak group, which is greater than 6 months. And at this meeting, there has been a lot of talk about the South African child who was treated for 40 weeks and still suppresses the virus for 8.5 years later. What is explaining this? Short answer, we don't know. Is it immunological control, that one or more of these bounce back or not, depending upon an immune function that we still have not recognized? Or is it something as simple as stochastic reactivation? Namely, the person has not gotten the right immunological stimulus, the right influenza or the right other infection that triggered virus? Or is it a combination of both? Again, the short answer, we don't know. Let's move on to therapeutic vaccination. Reviewing the literature of placebo-controlled studies that include interruption of ART to determine the efficacy of a therapeutic vaccine, no therapeutic vaccine has been successful in achieving durable suppression of HIV viremia. And so we decided we would give it a good shot. And we started a therapeutic vaccine study that we have just completed and submitted for publication. It's a study that we try to optimize for maximum effect. What do I mean by optimizing it? We used a vaccine that had multiple primes with DNA plasmids, multiple boosts using a VSV vector, multiple inserts as shown on this slide, including an adjuvant IL-12, and administered by electroporation. I don't think we could have tried any harder than that. We used subjects who were treated early in the course of infection, and we had a placebo-controlled group, 15 vaccine, 15 placebo. Here is the schematic diagram with the primes on the left, the plasmids containing multiple inserts, the boost on the right, and electroporation. And the therapeutic protocol was as follows. The arrows are the administration of the vaccine. Again, the blue is the plasmid with multiple inserts. The red is the VSV. They were given over a long period of time. This was an arduous study, 48 weeks of vaccination, followed by an interruption period. Now, I'm going to show you data with two types of endpoints. The first is the per protocol endpoint that triggers the necessity by protocol to restart therapy. And that is if you have 50,000 copy blip and it lasts for 4 weeks. But in addition, in order to compare our data to other studies that have been reported, we measured as a post hoc how long it takes to get to 40 copies and how long it takes to get to 400 copies. So let's look at the per protocol first. As you can see, very striking, there's no difference in the vaccine versus the placebo group for the median time to rebound. However, I want you to pay attention to the dark blue bars on the placebo group because a couple of them, including one in the vaccine group, went out for an extended period of time without rebounding, several hundred days to a couple of years. And I'm going to get back to that in a moment because I believe that's probably the most important thing that I learned from this study. What else did we learn? Therapeutic vaccine did not decrease the size of the reservoir. The vaccine group in red, the placebo in blue, multiple time points measured in weeks. There was no correlation between the size of the reservoir and the time to virus rebound following discontinuation of ART. Now, when we went back and said, okay, the per protocol is to start therapy when they reach 50,000 copies, is there any difference in these groups from the time you reach 40 copies of virus to the time you reach 400? And anyone can see from this Kaplan-Meier, there's no difference whatsoever. But here's the important thing. If you just look at the placebo group, forget the vaccine group for a minute, 20% of the subjects at 4 months had less than 40 copies. 27% at 4 months had less than 400 copies. And 40% at 4 months had less than 2,000 copies. This is important because there have been reports of studies that have not used a placebo which claim that there's an effect of an intervention when 30, 35, 40% of the people go out and don't rebound for X number of months. So this is very important because we're dealing with a moving target now, and we have to be careful how we interpret data. So quickly concluding this part of the talk, therapeutic vaccine did not prolong time to viral rebound. There was no impact on the size of the reservoir. There was no correlation with the time to rebound. But importantly, the study emphasizes the critical importance of placebo-controlled trials in assessing the time to rebound following therapeutic interruption. Now we'll go to the more complicated component of it, the last one on this slide, the passive transfer of HIV-specific antibodies. We're all familiar now with the HIV-native trimer of the envelope, which has a number of neutralizing epitopes. They've been identified by antibodies that have been cloned from HIV-infected individuals. Why broadly neutralizing antibodies? Well, they are very, very difficult to make in vivo. Humans have a lot of trouble. However, we have very little difficulty in producing these as monoclonal antibodies ex vivo from the cloned B cells of HIV-infected individuals. And so you might imagine there is considerable interest in the employment of the passive transfer of monoclonal antibodies for the prevention and treatment of HIV, and you're going to hear a lot about this at this meeting. So how do we get those antibodies? An HIV-infected individual has their B cells cloned. You make broadly neutralizing antibodies. We now have more than 200 of them. They're shown on this slide. You recognize some of the names already. They've been spoken about at the meeting. VRC01, 3BNC117, 101074, etc. On the right-hand side of the slide, there's 3 approaches for passive transfer. One is HIV prevention, and that's exactly what's going on right now with the AMP study, which is antibody-mediated prevention by repeated transfer of passive antibody VRC01 in a high-risk population. So we should have an answer to that soon. In addition, there's the question, does antibody directly suppress HIV? And the answer, it does. As shown from 2 of the studies, several have been published, one from Michelle Nutzenzweig on the left, and one from the VRC group at the NIH, showing that when you passively transfer antibody, on the left it was 3BNC117, and on the right it was VRC01, you can suppress viremia in a viremic patient. But what about the last one, the one that is the focus of our discussion today? Can you induce prolonged suppression of viral rebound following the interruption of ART? So let's get back to that yellow box. The answer to that is that you actually can. And let me tell you about some studies that are ongoing and some that will be done. Recently reported, one from Michelle Nutzenzweig's group and one from a combination study that was done independently but reported in the same journal from Pablo Tebas and my group at the NIH. We showed that when we passively transferred either 3BNC117 or VRC01, you delayed the rebound of plasma viremia following the interruption of ART. And if you just take our data, the historical time that I mentioned to you was around 11 to 28 days. We extended it, not impressively, but enough to notice, to about 39 days. Which brings up an interesting point. There are 2 possible scenarios that could follow this. One, the direct suppression of viral rebound requiring the continual presence of adequate levels of plasma antibody, which means that if we were able to successively give passive transfer of antibody, either alone or in combination, we could in fact have an ART-free sustained remission. You just need to pick what you want to be on. Do you want to be on ART every day, or do you want to have a passive transfer of an antibody intermittently? If the first yellow box is true, we can really amplify that. We can make more potent antibodies. We can extend the half-life of the antibody. Imagine giving an antibody once every 6 months subcutaneously. We can administer it by vector-based antibody production. And just like ARVs, we can give combinations. So I think that this is certainly a viable avenue to pursue. What about this? The induction of enhanced or distinct anti-HIV response that remains after the passively transferred antibody disappears. In other words, does passive transfer of antibody induce something that natural infection does not? We have a huge clue of that from a study from Mal Martin and Michelle Nutzenzweig that they published in Nature a couple of months ago. In a non-human primate, they showed that early antibody therapy can actually induce long-lasting immunity. So let me explain what they did. They took non-human primates, they infected them with Hiv, and they gave either a 2-week infusion of 2 antibodies, 3BNC117 and 101074, during acute infection. Then they waited until the antibody was gone from the system, and to their amazement, I would think, but great gratification, the virus did not rebound. They compared that simultaneously with infecting non-human primates with Hiv, treating them for 15 weeks during acute infection, stopping ART, and when they did, as you've seen in all of the animal studies, the virus came back. A very interesting finding. They went back and found out that when they did depletion studies, CD8 cells had been generated by that manipulation, which sustained the suppression of virus. a very interesting finding. What's the mechanism of that? Well, we don't know, but one of the possible mechanisms is since you're passively transferring antibody, that antibody may have complexed with either virus or a virus-infected cell, forming an immune complex that was presented to the immune system in an unusual way that the immune system did not see with the normal presentation of virus, leading to the HIV-specific responses that Martin and Nutzenzweig noticed. So let's get back to this box, and now what I want to do is insert in here something that we've been working on for the last several years. What about the passive transfer of an antibody against alpha-4 beta-7? For those of you not familiar about that, what is alpha-4 beta-7? It's an integrin present on leukocytes, particularly CD4-positive T cells. It has natural ligands shown here, and its principal function is to mediate migration to and retention of leukocytes in the gut. 9 years ago, Jim Artis and Claudia Cicala in my lab published a paper indicating that alpha-4 beta-7 was in fact a receptor on CD4-positive T cells that binds and triggers the cell. This is the schematic confirmation of the tall alpha-4 beta-7 closely juxtaposed to the CD4 molecule, ready to bind with the GP120 trimer. We went on to show that an alpha-4 beta-7 high cell is one that's highly susceptible to HIV infection. Shown very simply here, virus replication in the blue beta-7 high cells, very high, low replication in the beta-7 low, which is in red. So we asked the question, can a monoclonal antibody directed against alpha-4 beta-7 actually first prevent against transmission? This was a collaboration that we began several years ago with Tab Ansari, Francois Villager, Phil Santangelo, and a variety of others. And the work I'm going to show you now was done in their laboratory. They had a mouse antibody to alpha-4 beta-7, which they primatized to make it work in a non-human primate. I want to point out, and I'll get back to that in a minute or so, that there is also a human antibody that's actually on the market for human disease called vedolizumab, which is an antibody against alpha-4 beta-7. So what did we do with it? We used a transmission model in a non-human primate in which the monkeys received repeated low-dose mucosal challenge with SIV MAC251. And here's what we saw. Half of the monkeys did not get infected, as shown in this Kaplan-Meier plot, but even those who did get infected had a delay in viremia and much less virus in their tissue. So not only was it preventing infection, it was doing something in those animals that did get infected. So the obvious question related to what we're talking about today, an ART-free remission, was asked, can infusions of this antibody induce sustained virological remission following the interruption of antiretroviral therapy? And so we get back to the yellow box substituting passive transfer of anti-alpha-4 beta-7. We published this in Science in October, and what we showed that in animals receiving ART and antibody, there was sustained virological control. Very similar to what I just showed you in the Mal-Martin-Nutzenzweig. And here was the protocol. Animals were infected for 5 weeks. ART started on week 5 through week 18, overlapping with antibody to alpha-4 beta-7 that went through 18 into week 32, and then everything was stopped. And the results were striking. On the left-hand side, you see both groups of animals were infected. Both groups became aviremic on ART, but the control animals in red rebounded, developed viremia, and had to be sacrificed. The animals in blue, who received anti-alpha-4 beta-7, blipped a few times, but essentially went on to long-term control. And by long-term control, I mean now 1 to more than 2 years. And again, this was conducted in the laboratory of Tab Ansari. Now, we obviously wanted to ask the question, what is the mechanism of that? So we did cell depletion studies, and this is new data that has just been made by Tab Ansari and his colleagues. What we did was to take cell depletion with either an anti-CD8 alpha antibody, an anti-CD8 beta, or an anti-CD20. I'll explain in a moment what that does. Then we observed for rebound or not of virus, and then reconstitution of control or not when the cells came back. Now, if you look at cellular depletion, when you deplete with CD8 alpha, you deplete not only CTL, CD8s, but also NKs and NKTs. When you deplete with CD8 beta antibody, you deplete essentially only CD8 cells, because look at the NK, it's only 8.9. And again, B cell depletion doesn't deplete anything but B cells. And what they found was that if you look at the viral loads, when you get rid of T cells, CD8 T cells, but importantly NK and T, you get a blip of the virus back, and when the cells come back, the virus then gets resuppressed. No effect on the anti-CD8 beta, which only gets rid of CD8 positive T cells, and certainly no effect with anti-CD20. So what's the conclusion? The conclusion is that the durable control of viremia is due at least in part to a cell-mediated immune mechanism, and we're not exactly sure what that is yet. However, we have a hint, because the rebound of viremia and the return to suppression following cell depletion tracks very closely with the depletion and reappearance of NK and NKT cells. There may be, and it's likely, and we can't rule it out yet, that there is some contribution by classical CD8 positive T cells, and that cannot be ruled out. So we're seeing an interesting phenomenon where the original passive transfer of an antibody actually induces a durable response, and that's something certainly we need to pursue. Finally, and then at another experiment done by Cristina Guzzo and Paolo Lusso in my lab, they found an interesting unexpected finding, that when HIV buds off a virion, when HIV buds off a CD4 positive T cell as a virion, it incorporates alpha-4 beta-7 in its membrane. And I simply show it here. There's the T cell getting infected with HIV, budding off, and what you see is that it incorporates alpha-4 beta-7, similar to what we've known for a very long time, that when virions bud off, they incorporate DR molecules. Is this important? I don't know. What's the potential for it? It may be that part of the effects of antibody to alpha-4 beta-7 could be explained either by a direct antiviral effect, the formation of an immune complex that leads to a very interesting kind of antigen presentation, and or the blocking of homing to the gut. Now, for the last couple of slides, these are two papers from 2013. Vedolizumab, which is the human equivalent of the anti-alpha-4 beta-7 antibody that I was talking about in the non-human primates, has been successfully used to induce and maintain therapy and remission for inflammatory bowel diseases such as ulcerative colitis and Crohn's disease. So some questions arise. Is HIV infection in humans fundamentally different from SIV, SHIV in the non-human primate with regard to the role of alpha-4 beta-7 in disease pathogenesis? We don't know that right now. We want to find out. To observe a positive effect of vedolizumab in humans, will it be necessary to treat patients at a very specific time? Namely, do you want to treat them with very early in infection, and do you want to do it with vedolizumab, with ART, or with both at the same time? That's going to be an interesting question. If there's a positive effect of vedolizumab, what is the mechanism? And to answer these questions, you can only do that by doing a clinical trial in humans. And in fact, we have just started that, an exploratory open-label study of vedolizumab in subjects who are well controlled on ART for a considerable period of time. We have 15 patients in the study. This is the protocol, very similar to the animal protocol. As you can see, we're taking people who've been on ART. We give them multiple infusions of vedolizumab. We stop at week 22 ART. We continue the vedolizumab for 2 more infusions, and then we wait to see what happens. We don't have any meaningful results now, but we do have everyone enrolled on the protocol, and I would project that sometime in October, we'd start to get some hint as to whether or not we're seeing anything that's comparable to what we've seen in the animal model. And so, in summary, I've discussed the 2 major approaches that the field is pursuing towards a sustained ART-free HIV remission. What can I conclude? 1, the eradication of the reservoir is an aspirational goal that is highly unlikely to succeed on a large scale. Highly aggressive approaches, such as ablative conditioning and stem cell transplantation, are appropriate only for a very small subset of HIV-infected individuals with underlying disease that would require such an approach anyway. The preservation of natural immunity without any additional immune-enhancing approaches, as I described and as we've heard at this meeting, will prove to be effective in controlling HIV rebound following ATI for varying durations of time in a subset of people, like we've shown: Mississippi baby, French teenager, African baby. Therapeutic vaccines has not proven to be effective in controlling viremia following interruption of ART. The long-term passive transfer of combinations of long-acting, broadly neutralizing antibodies may be effective in replacing ART in certain patients. In other words, it may be one step towards an ART-free remission. And finally, the induction of durable HIV-specific immunity may be possible in association with passive transfer of HIV-specific or related antibodies, and a number of labs are actively pursuing this. Let me close by thanking a large number of my collaborators involved in this. I can't name them all, but they're on the slide for the therapeutic vaccine trial, particularly Mike Snella, Kat Claridge, Taeyuk Chung, and Susan Moya, as well as individuals in the HIV vaccine trials network, and of course our study participants. For the passive transfer of broadly neutralizing antibody, the Vaccine Research Center with John Muscola, Julie Ledgerwood, and their colleagues, our colleagues at the University of Pennsylvania, and in my own lab, again, Mike Snella, Kat Claridge, and Taeyuk Chung, and Susan Moyer, among others. And finally, the alpha-4 beta-7 studies, predominantly driven by the Tab Ansari Laboratory, by Francois Villinger, Phil Santangelo, and a number of others shown on this slide, and in my own laboratory, particularly Jim Arthus, Claudia Cicala, and Paolo Lusso. Thank you.